Today we start reproductive, and we're looking at male reproductive first. Male reproductive is more complicated than female anatomically just because there's more parts to the tubular pathway. So the male reproductive pathway, the whole purpose of course is to deliver sperm, and unlike the egg from the female, the sperm never leaves some kind of enclosed tubular structure until it leaves the urethra. In the female, the egg, once it leaves the ovary, can, by chance, move anywhere in the peritoneal cavity. It's only because the oviduct moves close to the ovary that it's taken up into the oviduct. So there's not a continuous pathway. And that's why we can have ectopic pregnancies outside of the oviduct, along the common iliac vessels or any lining of the peritoneal cavity is possible. Because the sperm can move out of the oviduct as well, so they can meet the egg anywhere in the peritoneal cavity. So our basic pathway here is the gonad, which is the term for both reproductive organs in male or female, is where the sperm have their stem cells, if you will, and they move through a series of ducts that we'll discuss, and they move into the head of the epididymis and down the body and reside in the tail where they develop chemical and physical abilities. So their structure is complete, but some of the enzymes haven't been activated yet. That takes place in the tail, and then upon emission, we're going to have three words that start with E, all right? Erection is the increase of blood flow to the penis, but that's not required for emission. It is required for ejaculation. So that's one E. Emission is the movement of sperm from the tail of the epididymis through the vas or ductus deferens and into the urethra. So once it enters the urethra, that's the end of the emission. We'll go through all of these in more detail. Emission is also the addition of secretions from glands, so seminal vesicles in the prostate gland and the bulbourethral gland. And then finally, ejaculation is both smooth muscle and skeletal muscle contraction and movement of the sperm and fluids through the urethral, through the penile urethra. Okay. So the names of those tubes and the histology of those tubes is a little bit more complicated than the female, where we basically have the oviduct, the uterus, and the vagina. Female, of course, picks up with it with all the hormones and regulation and all of that. So we do have hormones in the male, but they're not quite as complicated and covering as the female. Now let's look a little bit at development. Things have changed somewhat for Microsoft. I had a couple of movies here that don't work anymore. So I'll pull them up in just a minute. One's a clay animation of the development of male and female. So here's what used to be an automatic clicker question, but the software isn't connected anymore. We'll just look at it. The development of secondary sexual characteristics. You guys know what secondary sexual characteristics are?
Breasts or...
It's really the phenotype, okay, which is the appearance. So breasts, or the development of the scrotum, or the penis itself, or the clitoris in the female. So it's the appearance of being male or female. So when you go look at an ultrasound, and you can tell, at particular stages whether it's a male by the appearance of the penis. All right, so in the development of these characteristics, so the embryo will develop or will appear as a female. We're not talking about genetic changes. Those aren't going to change. Unless male genes present on the Y chromosome are expressed, or the embryo will develop as a male unless female genes on the X chromosome are expressed. A. All right? I haven't been able to find it, but when I first started teaching back in the early 80s, there was a film, literally a film, not a video, so on the real projectors, that was called The Fight to be Female. Actually, sorry, Fight to be Male. And it was exploring, actually, the concept of homosexuality. Not for lesbians, not what causes a woman to feel more like she was attracted to another female or feel male, but the male concept. And the premise that they tried to prove in the film was that male, and I'm a female, so I'm sorry, I don't know what that means, to feel male, but males need a exposure to a level of testosterone in the brain during fetal development to have the concept of being male. And they looked at a variety of situations. Rats that were male fetal rats that had had the testosterone decreased didn't act as aggressive as male rats that had normal amounts of testosterone. They went to England and interviewed a quote-unquote woman who was actually XY and didn't realize it genetically until she tried to have children and couldn't get pregnant and went to see a physician, and on examination she had undescended testes and no ovaries, and her genetic profile was an XY, but she felt like a female. Under British classification, though, with those chromosomes, she was really a male. It didn't go by, what she felt like. So the premise was that without the adequate exposure to significant amounts of testosterone, her brain developed as a female brain. They interviewed a true intersex, the old term hermaphrodite. True meaning it's like a chimera. The false are your XY, but you don't have sufficient testosterone, and so, like this woman in England, you develop looking to be female, all right? Or you're female, XX, but because of an adrenal tumor, you have too much testosterone, and so you develop secondary male characteristics. So that's the pseudo type of intersex. The true is one that has cells that are both. So half the cells in the body are XY, and half the cells in the body are XX. You have a testis, and you have a gonad, and you can have a penis, and you can have breasts. And they interviewed this person who, after reaching adulthood, was offered the opportunity to have surgery, plastic surgery, and she could choose to be one or the other. They would remove the ovary and reduce the breast to male size tissue, remove the mammary gland, or they could go the other direction, remove the testis, and surgically remove the penis, and still had a vagina and a uterus, so it had both organs, and the person chose to be a male, saying that that's what he has felt like all through development. Now, of course, in certain countries, it's easier to be male. There's more privileges to be male. So culturally, it's hard to say exactly what the complete influences were. But the most interesting, intriguing exposure in the film was an island in the Caribbean that they hesitated to, well, they did not give the location, where because it was small and an island, there had been some interbreeding among related individuals. And so many of the male babies were born with a deficit in an enzyme that converts testosterone to an enzyme that produces secondary sex characteristics called DHT, 1,2,4-dihydrotestosterone. And they didn't get sufficient testosterone produced to get converted to this until they reached puberty. So they were born looking like girls. And at puberty, which was around 10 or 11, their vaginas disappeared, the testes descended, and the labia fused to form scrotal sacs, and the clitoris enlarged to be a penis. So at puberty, the apparent girls became boys. And so they interviewed them to say, okay, was this, how weird was this? Well, my cousin did the same thing, so, but they had felt like they were boys anyway, so it was, again, that role of the expression of testosterone. So the presence, the expression of an SR gene on the Y chromosome needs to be present for that testosterone enzyme, for the testosterone and the DHT to be produced to form those secondary sex characteristics. So let's look first at what happens between male and female. We're looking primarily at male, and then when we start female, we'll compare that back again to the male. One of the things that you're going to need to do through the lecture, there's two things that I'm going to be emphasizing, is create a list of homologous structures. In other words, the testis in the male is homologous to the ovary in the female. Okay? Structures that are similar or developed from the same component. The other list is developed from. So in the sperm, There is a cisternae or a sac called the acrosome that contains enzymes that help the sperm penetrate its way through the egg protein coat. That develops from the Golgi apparatus. So one thing turns into another. Okay? So on your lecture exam, there will be a table where I will have you identify the origin, it develops from into something else, or its homologous structure. Okay, so as we go through both male and female, I'll be pointing out these characteristics. All right, so this sagittal view is looking at the posterior wall, the high abdominal wall of a developing embryo. And up near the diaphragm, remember where the kidneys are. Okay, we haven't done kidneys yet, but we did adrenal gland, which is just under the diaphragm. So up high near the diaphragm, we have the development of a pre-kidney structure, if you will. Now, without going into a lot of embryology, the kidney is unique in that three distinctly different organs appear. First one, and then it disappears, and as it's disappearing, the second one appears, and then it's disappearing, the third one appears. None of our other organs, we don't have one brain and then a second brain, okay? We have changes in the first organ that disappears, but not three distinctly separate ones. So the three different kidneys are identified as the pronephros, okay, the mesonephros, and the metanephros. This one never filtrates the blood. It never acts as a kidney. It functions basically to induce. You guys know what the word induce means? Embryologically, it causes changes. So as the optic nerve grows out from the floor of the third ventricle, it induces changes in the epithelia and the face of the embryo to invaginate and form the lens. If the optic nerve doesn't grow out, the lens doesn't form. So when we were talking about pituitary development, the outgrowth of the neurohypophysis induces the change in the roof of the mouth, the stomatidium, for the adenohypophysis to grow up. So chemicals are released in concentration gradients that cause changes in other cells. Okay, that's induction. So the pronephros, although it doesn't actually function as a kidney, appears to cause changes in the embryonic cells around it to form the mesonephros and the developing gonad. Okay? So it induces development of the gonad and of the mesonephros. The mesonephros does filter blood. So it does function as a kidney. And it does leave some remnants behind, okay, that form embryonic structures of the male reproductive system. It disappears in the female. And then the metanephros becomes the true kidney. So it becomes the actual functioning kidney. And we'll go through this again when we get to the urinary system. Part of doing reproductive system first, the last concepts that we cover are the first 6 to 8 weeks or so of embryonic development. And then from that, we can look at, not very deeply, but we can look at the embryonic development of all the other systems. We did that with the brain when we talked about the various vesicles of the brain. Okay? So This paramesonephric duct is a female structure that we'll talk about Friday, next week when we get to female. This is what forms female oviduct and uterus and the top of the vagina. All right? So paramesonephric duct is not present in the female. It disappears, as you can see here. It degenerates. And the mesonephros, there's the mesonephros there, it forms part of connecting tubes that connect the developing gonad to the duct system. So let me just move ahead a little bit to illustrate that for you. When we look at a diagram of the testis, here is the testis itself, right? And these structures right here, the efferent ductules, are part of the mesonephros that connect the testis, and then the rest of the mesonephric duct is going to form the epididymis and the vas deferens. So this is the mesonephros component right here, and then the mesonephric duct is going to form epididymis and ductus deferens and the top of the urethra. Okay. So this is what's going to form part of that tubular structure. Now the testis develops right next to the developing kidney. And that's why they share in the male this genital, sometimes called the genital urinary system, where the sperm travel through the urinary pathway in the urethra because of that close component of development. So we'll look at a little video of this duct system. But the testis, as I'm indicating here, develops up high on the posterior abdominal wall. Where does it end up? Down in the scrotal sac. All right? So as the testis is developing along the posterior abdominal wall, there is a band of connective tissue that attaches it above, and a band of connective tissue that comes all the way down to the scrotum. This band is known as the gubernaculum. Testis. Okay. This is the upper. And this is the lower. The upper disappears. In the female, it remains and becomes a ligament between the ovaries and the uterus. The lower gubernaculum shortens. It's attached to the scrotal sac, so as it shortens, it's going to pull the testis down into the scrotum. This happens during fetal development, generally by the 12th week or so, or starting to pull down, and at 7 months is typically of gestation, not 7 months after birth. It's down in the scrotal sac. A baby may be born with one or both testes undescended. This is typically not a problem unless they remain undescended into puberty, because the developing sperm undergo mitosis and produce more and more. High body temperatures can result in this not happening correctly, and infertility as a result. So surgical descent of the testis is required prior to puberty if they don't do it on their own. So you have this diagram, I believe, in your lecture notes. So this large orange structure here is illustrating the mesonephros. So this is the kidney that disappears. The paramesonephros, or rather the pronephros, starts up here and then starts, as the mesonephros appears, the pronephros starts to disappear. So they kind of overlap a little bit. So this is a partially functioning kidney, and the true kidney is going to start to grow up here. But as this disappears, see these ducts that are connecting it to the mesonephric duct? These really short ones right here? Those are what form connections from the testis to the mesonephric duct. So I'm going to write this in a different color. The mesonephric duct remains, all right. Part of the mesonephros remains as etheric ducts. And the mesonephric duct remains and forms the epididymis, the vas deferens, and the upper portion of the urethra. Essentially where it passes through the prostate gland. So that's this brown structure right here. So we have the efferent ductules from the mesonephros, and then the mesonephric duct forming the epididymis and the vas deferens, and then a portion of the urethra. When we get to the female, that disappears, and a different duct structure develops, which disappears in the male. So the paramesonephric duct is going to disappear in the male. So here's an image of the descent of the testis. So this band right here is the gubernaculum, and it's not showing the upper gubernaculum. I'm only mentioning the upper gubernaculum because it remains in the female, and I'll come back and talk about it when we get to the female. So the lower gubernaculum passes into the scrotum, and as it shortens, it pulls the testis down. This is behind the peritoneum of the abdominal cavity. So the peritoneum is the membrane that lines the abdominal cavity, covers the organs, so it's behind that. As it pulls down and passes through the inguinal canal, it pulls a series of layers of muscle and connective tissue with it, as well as peritoneum. So that's going to bring us into the context of discussing the layers of the inguinal canal. And then this is just a frontal view as it moves down. All right. The female gonad, the egg, does not need to do that because all of the mitotic divisions, daughter cell formation, occurs before the baby girl is born. They occur in the male and then stop, and then at puberty restart again. So we're born as females with all the eggs we're ever going to have. Whereas in the male, they kind of have stem cells and they can produce millions and millions and millions more. So let me see if I can pull up this video. It's in French, so I don't really have the sound on. My French is quite old. Just let it play, and then I'll stop it and kind of explain what's going on. start this. So what you're seeing here is the pronephros. Okay? So this is that early kidney that really doesn't function to do any filtration of blood. It doesn't create any filtrate, but it induces the development of this mesonephric duct. This is what's again going to become the tubular structures that the sperm passes through. All right, now as the pronephros is disappearing, these structures belong to the mesonephros. So these are the early filtration structures that are appearing, that are going to filter the blood, and these continue to develop from the mesonephric duct as the pronephros disappears. Now the pronephros is gone, and we have the mesonephros with the filtration structures growing out of the mesonephric duct. Again, the green thing is the mesonephric duct. So now the orange is the mesonephros. Right? So we have the mesonephric duct. This is our first functioning kidney. And in the next set, you'll see the gray gonad or testis appearing, induced by the developing kidney or mesonephros. The brown structure at the bottom, reddish brown structure, is the bladder. So down here now at the bottom, we're starting to see the metanephros. So it develops separately, actually starts down near the bottom and moves up the posterior abdominal wall. These are the ureters that are connecting the flow of urine to the bladder. So again, the urinary bladder, the true kidneys, the metanephros, our mesonephric duct, and then in just a moment you'll see the gonads developing. So up there you can see a little bit of the gray gonad. The blue is the paramesonephric duct. That's going to be a female structure. So it develops and then disappears. And the females will see this will disappear as the blue remains. So here's our growing true kidney. Our mesonephros is disappearing. Our gonad is being pulled down towards the pelvis by the gubernaculum. Here we're seeing the connection between these are the efferent ducts that maintain the connection between the testis and the epididymis, part of our mesonephros. So we have a testis, we have the efferent ductules, we have what's going to form the epididymis, vas deferens, and connect into the bladder, into the forming the urethra. Okay? And so we have that. I don't know if I still have this up as a link from last year or not, but if I don't, I'll put it up on D2L. So we'll watch another one that is the female. Okay, you can see how the mesonephric duct disappears and the paramesonephric duct remains. So those are they're not the same structure, but they're homologous. So the paramesonephric develops into the duct structures of the female. Mesonephric develops into the duct pathway structures of the male.
Can we go back just really quick and look at the title of the French show?
Oh, yes. It's urogenital animation. Male urogenital development. I'll put a link up on the announcement section. All right, so we have a series of muscles that we're going to learn to identify. Reproductive are these two right here. These are in the male. This is actually a urinary bladder one. We typically don't name smooth muscles, but there's 3 or 4 that we do. So these are smooth muscle. This one is skeletal muscle. All right. So let's look at how these first two are formed as a result of the descent of the testes. So this is adult. This is mature. And we're going to be looking at the layers of the coverings of the scrotum. So the scrotum is the sac that contains the gonads, the testes. The spermatic cord is formed of the blood vessels, nerves, and vas deferens. that link the testes to the pelvic cavity. So remember, the testes descended. They're supplied with blood vessels that come off of the aorta. And they come off of the aorta relatively high. When we do cardiovascular system, I'm going to go now. Vessels. But it makes sense about how high they actually come off. So here's the diaphragm, here are the kidneys and adrenal gland, and this is where the gonads start their development. Here's the gonadal arteries. So in the female, they come down to the pelvis where the ovaries are found. In the male, they actually pass through the inguinal canal and down into the scrotum. They start up relatively high in relationship to where the gonads are because of that site of actual embryonic origin. So as the testes are pulled down into the scrotum, they have to pass through the lower abdominal wall. So they're still connected via blood vessels, and the veins have to return. We have the vas deferens, and we have some nerves. So all of those form what's known as the spermatic cord. Okay. As such, I don't ask you to identify in lab the spermatic cord. It's too difficult to kind of get the idea of the content in a lab model. But for lecture content, you should be able to identify the structures that form it, and it makes sense. It's whatever we need to carry blood to and from the testes, and to carry sperm away from the testes, and to innervate. So that's what forms the content. But as the testes are pulled through, we start pulling through layers of connective tissue, layers of muscle, layers of fascia, and the peritoneal membrane that is attached to the posterior abdominal wall. And that creates the coverings of the spermatic cord. So this is a picture of one of our models. And we've got those coverings and the content here. All right. So let's look at how this is formed. Which is showing the same thing in a cat dissection. So we have the abdominal content, spermatic cord with the layers removed, and then the testis with the scrotum over that. So here's the lower gubernaculum, and we call the passageway through the abdominal wall the inguinal canal. You've probably heard of an inguinal hernia, right? By creating this passageway, there's an automatic weakness in the abdominal wall. If that weakness is enlarged because of straining, lifting heavy weights, constipation, and having a hard time having a bowel movement or whatever, then that's what we call an increase in that weakness. An opening is called a hernia. So babies can develop umbilical hernias after they're born because in embryonic development, when the embryo folds, there's an opening around, it fuses around the umbilical cord. The abdominal contents actually move out of the fetal development and develop outside, and then come back in and twist and come back in, the small intestines. And so at that site where the umbilicus is and where the abdominal wall is closed, there's an inherent weakness. Babies cry a lot. That can enlarge the separation between the two halves of the abdominal wall, can enlarge, and you could have a small 1 to 2 inch swelling where small intestine passes between the fusion between the two rectus abdominal muscles. You could have a weakness in the diaphragm where the esophagus passes through, and the stomach or intestines can push up into the thoracic cavity. Okay, so hernia is just that weakness that develops in the abdominal wall. So a hernia can develop associated with this channel known as the inguinal canal. It's angled, men and women have it. All right, so we don't have, as women, we don't have the vas deferens. We don't have the blood vessels. What would we have as women passing through the inguinal canal, thinking about what we just talked about so far? What structures, of all the things I described, would a woman have in female development? So in the male, notice the lower gubernaculum is what pulls the testis through the inguinal canal to the base of the scrotum. In the female, the lower gubernaculum is a ligament that attaches to the uterus, passes through the inguinal canal, and attaches to the labia, the fold of lips on either side of the vaginal opening. So we have that structure passing through the inguinal canal. It's not very large, so it's very uncommon for women to get inguinal hernias because we don't have as much material passing through, but we do have some, okay? So as the testis is pulled through, as I said, it's going to pull down layers of fascia and layers of muscle and peritoneum. So I'm going to erase the board in just a second and diagram this for you. This outer layer out here is showing the skin of the scrotum. This inner layer green is showing the peritoneum. Now we haven't talked about peritoneum since the first week of 4:30. Anybody remember what a serous membrane is? Remember we had 4 different types, and we have 4 different types of membranes? We have, skin is one, and synovial membranes, lining the mouth, lining the respiratory tract. Depending on the urinary tract, we have different epithelia, and we have mucous membranes. So a membrane with a layer of epithelium plus connective tissue, loose connective tissue. A serous membrane has simple squamous epithelium. So it's producing a watery fluid, and organs like the lung grow into it like a sac. And we have the visceral membrane and the parietal membrane. The fluid between creates lubrication. So in the abdominal cavity, may I erase this part of the board? You guys have that down already? It's been up for a while. We look at a sagittal view of the body. Here's the diaphragm and the bladder and the rectum. So what I'm drawing here is the appearance of a continuous sac that's secreting serous fluid on the outside and has connective tissue attaching it to the body wall or to an organ. All right, so think of a plastic bag that's water inside it. Now, an organ such as the liver grows into this continuous bag. Now, the connective tissue attaches it to the organ. Well, out here it's attached to the body wall. So, if this is the lung, this is visceral pleura and parietal pleura. We're in the abdominal cavity, so we have visceral peritoneum attached to the liver, but we have parietal peritoneum attached to the body wall. Right? Now the kidneys are attached to the back of the body wall, so the parietal peritoneum passes over the front and doesn't pass behind it. So we call this retroperitoneal. Well, guess what? If the testes are also retroperitoneal, and they are being pulled down into the scrotal fat along this pathway, they're always behind the peritoneum. Okay? So this is that parietal peritoneum. And we'll see how that makes a difference and what layers get pulled down. So the testis is being pulled down here, and as it passes through the abdominal wall, it's going to take these layers of organs with it. So let me show you a diagram from Netters to kind of explain that sequence. So this is the parietal peritoneum. In the front, we have the anterior abdominal wall. And we have the rectus abdominis here. This muscle would be what? External oblique, internal oblique, and transverse abdominis. All right. Now notice that the external oblique muscles stop and their fascia becomes a tendon that blends into, remember the semilunar? Any semilunar lunaris, so it's going to blend into that and onto the pelvis. Same with the internal oblique and transverse, and here's rectus abdominis. All right? So this passageway right here, more lateral posteriorly, or deep, more anterior superficially. This passageway right here is the inguinal canal. And here's the spermatic cord. So as it comes through, these would be the testicular vessels. It's going to pull layers of the superficial fascia. It's going to pull layers of the fascia of the transverse abdominis. It's actually going to bring with it some of the muscle fibers of the internal oblique. And the fascia of the external oblique curves under, forms the floor, the roof, and the outer wall of the inguinal canal. And also gives layers to the spermatic cord. So it's kind of like setting up a series of building blocks. All right, and this creates a channel that is angled. So if this is the external abdominal oblique and the internal abdominal oblique and the transverse abdominis and fascia, and then we have the peritoneum. So that's what we see right here. Okay? So if you have that diagram in your lecture notes, so you can use that to follow along while I diagram this. Need some different colors. So we have the skin. This is going to be the scrotum. And as you learn with skin, there is a connective tissue deep to it, all right, our papillary layer and our reticular layer, and then some adipose tissue. And then just deep to that is connective tissue that blends into the muscle. So in that connective tissue deep to the epithelium of the skin, we have smooth muscle. Muscle is identified as the dartos. It's under influence of the autonomic nervous system, sympathetic nervous system, which goes to the skin. And when an individual, male obviously, is cold, it causes the skin of the scrotal sac to wrinkle, contract, and become wrinkled. Now the next layer that you'll see is the external abdominal oblique muscle. So it's muscle out here. But it doesn't contribute muscle to the spermatic cord or to the scrotum. It contributes a layer of fascia. So this layer is known as the external spermatic fascia. It's around the spermatic cord, but it also forms the covering and it forms the floor of the inguinal canal. Okay, deep to the external spermatic fascia, we have the the external abdominal oblique, we have the internal abdominal oblique. And it contributes skeletal muscle fibers. So skeletal muscle fibers of the external abdominal oblique are given a different name. This is now identified as the cremaster muscle. Remember I told you that you would see questions that say, this develops from what structure? So the external spermatic fascia develops from the external oblique muscle. The cremaster muscle has its origin from the internal abdominal oblique. Further develops into that. Then we have the transverse abdominis. And that doesn't really contribute much. And then we have more connective tissue, which is the internal spermatic fascia. I'm not going to hold you responsible for that. It comes from a band of connective tissue called the transversalis fascia, which we really haven't discussed a lot. So we're going to skip this third one here, or the fourth one, the internal spermatic fascia. And then we have the peritoneum. Now remember, all of these surround the testis because it's passing through the wall. So if I have a bunch of sheets hanging over a clothesline, and I decide to walk under that clothesline, I'm going to pick up each of those sheets that are hanging over the clothesline, right? Because I'm passing through. So that's how each of these forms a layer around the testis. However, the peritoneum is already behind it. Okay, so it's not passing through it. So imagine you're sliding down this wall, this slippery wall, and you've got this layer of this tarp that's over you. So as you're sliding down, you grab onto it, and then you slide into a well. You're grabbing onto it. Is it going to be around your feet? No. No, it's going to be double layered in front of you. All right, so as the testis comes down here, it grabs onto the peritoneum, and the peritoneum is pulled down into the testis, down into the scrotal sac, in front of the gonad. And so this structure up here is peritoneum. But down here, we call this closed little sac the tunica vaginalis. Not only do you guys have one vagina, you have two. So that's this structure right here. Okay. It tends to be collapsed and fused, so it's not really connected to the peritoneum. But sometimes that's open, almost like a cyst, and that tunica vaginalis can contain fluid and create a swelling on one side of the scrotum. It's called a water sac or a hydrocele. So if this enlarges, it forms a structure like that, and it can be surgically just kind of like drained. Okay, a needle can go in there and drain the fluid. But parents get concerned if one side of their baby's scrotum is larger than the other. So that's what you're seeing here. Yes?
So as the testes goes down, does it like create its own hole in a sense? Like
Well, the hole is already
there?
So think of the labia in the female, which are the labia majora, where the hair follicles are this kind of already created, slight, widely open pouch of skin, and the scrota, the gubernaculum, is attached to that. So the opening through the abdominal wall is already there because of the gubernaculum as the embryo folds and fuses, and the muscles develop around it. Then as the testis is pulled through, it enlarges that. Okay? So here is a hydrocele, okay, where there is a connection from the tunica vaginala to the peritoneal cavity and can create this water space, water fluid-filled space. All right, so without looking at the diagram, because the answer's on the board, the cremaster muscle develops from which of these up here? Internal abdominal oblique. Okay. Now it's skeletal muscle. However, it is controlled by an autonomic reflex called the cremasteric reflex. It can be triggered in guys by just kind of tickling the side of the upper thigh, inner thigh. It's also a response to cold. And notice how the cremaster muscle goes all the way around the testis and the scrotum. So when it contracts, it actually pulls the testes up closer to the body and sometimes into the inguinal, partially into the inguinal canal, so that can be pretty uncomfortable. There's a classic Seinfeld show where, what is his name? Charles? No. George comes in from swimming, and he's explaining the change in his appearance due to the cold water from going out swimming in the middle of the winter time. So both the cremasteric reflex and the dartos muscle keep the testis warm. All right? We'll look at some other structures that keep the testis cool, as that's actually just as important or more important. So again, back to this diagram, based on what we're looking over there. Here's the external abdominal muscle, so oblique, contributing fascia to form the roof and floor, as well as the wrapping of the spermatic cord, the external spermatic fascia. Carries the internal abdominal oblique, contributing, it's arching over the spermatic cord, but contributing skeletal muscle. The transverse salus muscle, the transverse salus fascia, that's what gives rise to what we're not talking about, the internal spermatic fascia, and then the peritoneum that gets pulled in as well. Now this is what the abdominal wall looks like from the inside. So we were looking at it from the external view. This is looking at it from the internal view. Typically gets cut off when we do the remove the front here. We're not quite far back. So this white structure right here is the vas deferens. Okay. Here we have the ovarian artery and testicular vein, and those are also going to pass through this opening. So this is the location of the deep inguinal ring, sometimes known as the internal inguinal orifice. So the external is out here, and it passes at an angle to get to that point. And then the vas deferens comes into the, wraps around laterally and posteriorly to the bladder. Now, let's talk about an inguinal hernia. So here's the rectus abdominis, linea semilunaris, transverse salus fascia, the transverse abdominis muscle. This would be the pelvic bone and the inguinal ligament with the femoral artery and vein passing underneath it. All right, here's obturator artery passing through the obturator foramen. And here we have the vas deferens and testicular blood vessels coming down from the aorta going up to the inferior vena cava and renal vein. Now, the area of weakness is typically right here. This is the most common. These blood vessels are called the inferior epigastrics, and they are a marker for whether it's an indirect or a direct hernia, which at first definition seems to be backwards. All right? So remember our inguinal canal is kind of going at an angle like this. The deep ring is more posterior and lateral. The superficial ring is more anterior medial. So if we have pressure from coughing a lot, or as I said, lifting the improper way, not using your legs, but using your abdominal muscles to strengthen that, this area of weakness opens up, and a loop of intestine can enter the inguinal canal from the posterior aspect without going through the opening. Okay? Medial to these blood vessels right here. That's known as a direct inguinal hernia. If instead a loop of intestine, this gets enlarged and the loop of intestine actually passes through the opening, that's called an indirect, which to me always seemed backwards. Like the easiest way seemed to be to go through the opening, but it's not the straightest route. All right? So if I wanted to leave the classroom and I walk straight through here, it'd be really difficult, and out this wall, I have to push through the wall first. So that's the most direct route. So that would be the direct route of a direct inguinal hernia. If instead I came around and went through the doorway, well, that might be easier. That takes longer. It's not a straight pathway, and that's an indirect route. All right, so that would be the indirect hernia. So direct hernia is medial to these inferior epigastric vessels, and the direct is lateral, passing through the opening. If we go back to this image, it might clarify it for you a little bit. Here's those inferior epigastric vessels I was talking about. So a direct hernia is going to come right through here and come into the posterior wall of the inguinal canal. And have a loop of intestine. Both of them can come out the superficial ring right here. Indirect would come through here and then pass into the inguinal canal. So they both go into the inguinal canal. One passes through a weakness in the posterior wall. The other passes through the actual opening, the deep ring. Okay. So here's a direct. This is the deep inguinal ring right here. But instead of entering that, the loop of intestine is passing, creating a separation of these layers and coming into the inguinal canal from the back. Okay, so a straight, think of direct as the straightest route, the shortest distance between 2 points. And here's the indirect. Notice how it's lateral to these inferior epigastric vessels and comes into the inguinal canal along with the vas deferens and vessels. Typically, this becomes noticeable in the individual when they cough. It's not necessarily painful, but they'll feel a swelling that can usually be reduced by putting pressure. Reduced means they smaller and disappear by putting pressure on it. Okay, so the guy will, for a while, will just, every time he coughs, will just kind of hold that area. Finally, that will be swollen all the time instead of just when they cough. And the main problem is if there's a twisting in the intestine, it can cause blockage or gangrene as it loses its blood supply. All right, so that's the major reason for doing surgery. It's not discomfort, but to prevent complications. All right, so with that definition, this should be pretty clear. So A or B is a definition of a direct inguinal hernia. B. Which one? B. Yeah. Okay, this is a good place to take a break a little early, but concept-wise. So just take a short break, 10 minutes, come back at about 1:15. And an adult testis, it's about the shape of an egg. We have some plasticized ones over here.
She already started?
No, she didn't. So these are fixed and then embedded with a plastic that hardens. And so this would be the testis. The layers have been cut and pulled away. And here's the spermatic cord. So this structure is a blood vessel, and then this larger central one is the vas deferens. And you can see some faint pink fibers, and those are the cremasteric muscle. Okay, a little bit more defined here. So the larger structure is the vas deferens. So when we look at the, this would be the epididymis. coming down here and coming back up. And so the testis itself is covered by a band of connective tissue. I'll leave this diagram on the board here for just a moment. I should have moved it over to one side. So we have this band of connective tissue giving a white coat to the testis, known as the white coat. Kind of like the orange peel or lemon. And then the pith of that separates the citrus fruit into segments. And so we have bands of dense irregular connective tissue penetrating into the testis and forming lobules, okay, or small segments. Within each of these lobules are about 15 to 20 seminiferous tubules. The seminiferous tubules are the tubular structures from within which the sperm actually develops. So we're going to look at those in larger detail. I'm just going to draw a couple here. They are highly coiled. And they come together and join to form a tubular structure known as a straight tubule. So this would be the seminiferous tubule. This is the straight tubule. If you want to keep using Latin, you can call it tubuli recti, recti meaning straight. All right, so we're at this point right here. And then a variety of all the straight tubules will come together. And will form a kind of maze right near the edge of the testis, this region right here. And that's this area right here. And that's known as the reedy testis. So straight tubules join together to form the reedy testis. And that's still within the testis. Okay, it's still part of the gonad. And then the reedy testes will form several connections to the epididymis. And these channels are the efferent ducts, or efferent ductules, that are the remnant of the mesonephric ductules of our mesonephros kidney. That's why I left this up. So in the embryonic development, remember we had the pronephros, and then these filtration structures and mesonephric tubules developed attached to the mesonephric duct. They have these kind of cup-like structures that you saw in the video. Those connected to the developing testis and remain when the rest of the pronephros and mesonephros disappears, connecting the testis to the mesonephric duct. All right? So here is the testis. Here are those mesonephric tubules remain, and they are identified as efferent ducts or ductules. And that is these individual structures here. So that is everything in blue and black is the testis or gonad. The green is our embryonic structure that was the mesonephros and its duct. So mesonephric tubules and now the mesonephric duct forms the epididymis, which is a single, highly coiled tubule that we'll get to in a moment. Actually, I should draw that in red.
Did you
Because
say
that's coming from
the efferent ductule is remnants of the mesonephric tubule?
Yes, these structures right here.
Okay.
So epididymis is part of the mesonephric duct. Okay. Again, on this, here's the pronephros that disappears. Here's the mesonephric duct that we want to become the epididymis. Here's the beginning of the mesonephric tubules that are forming the efferent ductules. And they have connective tissue around them, which is what we see there is the mesonephros. Inside that are those mesonephric tubules. So our pronephros is gone. The mesonephros is growing closer to the bladder. We won't worry about the development of the true kidney because that's really, here comes the gonad. And it's making connections with those mesonephros tubules as the mesonephros structure itself starts to disappear. So in here, we're seeing the connection between the mesonephric tubules and the gonad. These structures right here are the efferent ductules. This is going to become the epididymis and then the vas deferens, because all of that is connected. So here's efferent ductules. Okay. So there is a continuous closed pathway that the sperm pass through. So seminiferous tubules, straight tubules, reedy testes are components of the gonad itself. Efferent ductules, remnants of the mesonephric tubules, all of these here, drain into the epididymis, which is part of the mesonephric duct. Now, before we look at the detail of the seminiferous tubules, the testis is supplied by blood, which is from the testicular artery, and drained by the deoxygenated blood with the waste products, carbon dioxide, and the waste products of metabolism are carried away by the testicular vein. However, you'll notice in this image that the testicular vein is not a single structure. It's reticulated. There's a network of blood vessels that wrap around the artery. You can see that as well, a little bit better in this image of the model. So here we have the testicular artery, and then we have this network or reticulation of the testicular vein. As such, that is called the pampiniform plexus. So the testicular vein develops into, remember that table I said, that list of terms, things that become something else or are the origin of something else, like the internal abdominal oblique is the origin of the cremaster muscle. So here the testicular vein within the spermatic cord and scrotum become a network of veins known as the pampiniform plexus. If you were to identify it as the testicular vein, you wouldn't be wrong, but you wouldn't be fully correct. You wouldn't be accurate. So I wouldn't give you full credit. It would be partial credit for that. All right. The reason for this, again, is temperature control. So remember the cremaster muscle pulling the testis closer to the body and the dartos muscle wrinkling up the skin help to keep the testis from getting too cold. But just as important is to keep the testis from getting too hot. So core body temperature is too hot for normal sperm development. Well, guess where the blood, the arterial blood, is coming from? It's coming out of the abdominal aorta. So it's carrying heat, too much heat, out to the testes. So we have a counter-current flow of hot arterial blood coming into the testes and cooler venous blood leaving. So the heat in the arterial blood is transferred to the venous blood, and because it's counter-current, as the venous blood is warmed, moving closer to the body, it's coming in contact with hotter arterial blood that has just left the abdomen. And as that arterial blood is cooled, moving down the spermatic cord, it's coming in contact with always cooler venous blood. So there's always a temperature difference. And there can be a transfer of heat from the arterial blood to the venous blood. We see this in cold water mammals. So whales with their tails, their flukes, are very thin compared to the rest of the mass of the whale. And out in the Arctic, where the water is frigid, they could lose a lot of body heat across that surface area. And so they have a similar process where hot arterial blood from their abdominal cavity, as it moves out to the fluke, is cooled by the incoming very ice-cold venous blood. So we're not bringing cold venous blood that takes up body temperature to warm it up, and we're not losing that heat to the water. It's being transferred to the venous blood. So the same principle is applied here, and that functions to keep the developing sperm cells at a cooler temperature as they require. So that is a cooling effect, whereas dartos and cremaster muscle are a warming effect. So let's look at the structure of the seminiferous tubule. Okay? Remember I said that this is where the sperm, the stem cells of the sperm are found, and where they are going to develop into their mature appearance. They don't develop the ability to actually propel themselves until they reside in the tail of the epididymis, but they look mature by the time they leave the seminiferous tubule. May I raise the board? Okay, back. You're going to have more cross sections than you have individual pieces of spaghetti because they coil back and forth. So this image right here is representing one cross section through a tube, seminiferous tubule. Right? In the microscope, you're going to see multiple cross sections. And these may be two different seminiferous tubules, or all three of them may belong to the same one. You can't tell. They have a lumen. So without drawing in the developing sperm, the wall of the lumen is formed by simple columnar cells, kind of pyramidal. And these are known as Sertoli cells, which is the older name, be an eponym. Or sustentacular cells. They're epithelial. They're actually a form of simple columnar epithelium. Okay? Their nucleus is quite distinctive. It's about the only part of the sustentacular cell that is identifiable. It's kind of an angular cell. You remember mast cells from last semester? How they kind of were angular. They look, the nucleus kind of has the shape of a mast cell. They're not orange. So that would be the nucleolus, and that's the nucleus of the entire cell. Now the reason I'm drawing them first is because we see a whole bunch of other nuclei in cells when we look at a histology preparation. All of these other nuclei tend to be round, and these are the developing sperm. This is actually the nucleus of the sustentacular cell. Okay. We'll get to the appearance of that in a moment. There is a connective tissue capsule. Loose irregular first and some dense irregular, because we always have loose irregular deep to an epithelium. And then in between the seminiferous tubules, we have clusters of other additional epithelial cells that are endocrine. These are the cells that are going to synthesize testosterone. All right? And these are identified as interstitial cells. The older term are interstitial cells of Leydig, and now they're often just referred to as interstitial cells. So this whole diagram is of seminiferous tubules. So the sustentacular cells are sometimes called nurse cells because they form a supportive function in the development of the sperm. They release a hormone. They themselves can synthesize a hormone. And they bind androgen, they bind the testosterone in high concentration to keep it close to the developing sperm so it doesn't all just dissipate in the blood vessels. So now let's take a closer look at how these sperm appear. So this is the outer part of the one seminiferous, one sustentacular Sertoli cell, and this is where the lumen is. Okay? I actually prefer to flip it around and draw it the other way, so bear with me. So here is the basal membrane, the bottom membrane. Here would be the connective tissue of a sustentacular cell. And outside that cell, so between the connective tissue, is our stem cell for the development of the sperm. So it's intimately associated with the plasma membrane, but outside the sustentacular cells. It's not in contact with the cytoplasm. The best correlation I can provide you for this is to remind you of how Schwann cells wrapped around unmyelinated axons. So remember we had the Schwann cell, and we would have cross sections of axons in here. Not multiple wrappings, but the axon would have a single layer. It would be inside the Schwann cell, but it would be kind of like you push your finger inside rising bread dough. Okay, be enveloped by it, but not actually inside the cell. So that's similar to the relationship of our developing sperm and the Sertoli cell. So this first cell is identified as a spermatogonia. That is what would be present in the testis of a newborn male child. Right? They would all be spermatogonia. Spermatogonia are diploid cells. You know what that means? They're like every other body cell in your body. They have the two-end appearance. So one end are the 23 chromosomes that were in the sperm that created you. And the other end are the 23 chromosomes that were in the egg that was fertilized by the sperm. So each sperm and each egg, before they're fertilized, are in. And when the two come together to form a new cell, it's 2N. So N represents the 23 chromosomes that came from each of your parents. All right? So each of our body cells, muscle, nerve, epithelial cell, contains 46 chromosomes, 23 pairs. half of each pair came from your mother, the other half came from your father. So in this spermatogonium, half of the sperm came from this guy's mom, and half of the chromosomes came from this guy's mom, and half came from his dad. All right? So we call this 2N. That's considered to be diploid, when you have representatives of both parents' chromosomes. And there will be several spermatogonia along the outer edge between the connective tissue of this sustentacular cell. And that's the way it remains through childhood until puberty occurs. So on this diagram, these would be the spermatogonia. Okay? They're always at the edge, just along the connective tissue. And they're slightly large cells. They're not the largest, but they're the second largest, if you will, of the cells that appear. And I'll show you a micrograph in a moment. Now, under influence of testosterone, once puberty occurs, and this is continuous, the testosterone coming from the interstitial cells, some of the spermatogonia are going to undergo mitosis, the typical cell division of any cell in the body, right? Producing 2 daughter cells. You guys familiar with mitosis? So 2 daughter cells are formed. One remains behind, or 2 son cells, if that makes more sense. One remains behind as a spermatogonium. The other one moves up into the cytoplasm, not actually into it, but up towards the lumen. And so always outside the cytoplasm, always outside the plasma membrane. So this is going to move up here. And again, still surrounded by a plasma membrane, so still outside the actual cell. It doesn't penetrate that plasma membrane. And it hasn't, this would be one daughter cell, and this would be the other daughter cell. Okay? So this remains behind as a spermatogonium, and this one starts to undergo meiosis, where we start reducing the number of chromosomes. To do so, it will replicate its DNA again, and the chromosomes are duplicated, replicated, and become quite large. And this is the largest of the cells that we see. So really the only difference so far is the other daughter cell, but now we've replicated the chromosomes again, and they've tightened tightened down so we can actually see the chromosomes. And so this cell is identified as a primary spermatocyte. I'm going to go through these stages, but I'm going to wait until Thursday to actually talk about meiosis, okay? Because I want to get more into the anatomy structures so you can look at some more things in lab today. Now, the primary spermatocyte, it's duplicated its chromosomes. Now it's undergoing to go the first of 2 cell divisions. So it divides into 2 cells, which are still attached to each other by cytoplasmic bridges. Our primary spermatocyte, by the way, was still a diploid. We're going to, again, come back in detail. Our primary spermatocytes now have duplicated, but not in mitosis. So their separation of chromosomes is no longer the same. All right? So these are now haploids. This is a secondary spermatocyte. We because these are duplicated, I actually write this as 2N. Don't worry about it now, I'll explain why on Thursday. These have divided, so they're now just a haploid, but they have 2 copies of each. So if there's a Y, there's 2 copies of the Y, okay? So let me just briefly, again, I'll go back over it on Thursday. A diploid would be an X and a Y. X from the mother, the Y from the father, okay? Now when those are replicated in the primary spermatocyte, we have an XX and a YY. So it's the 2N is X and Y. Here we have an XX and a YY, so it's 2 times the X and the Y. Does that make sense? If it doesn't, just wait till Thursday, okay? So now this is split. N is just going to be X, remember, or Y. But remember, we replicated it. So now it's 2X or it's 2Y. Okay? So it's not really diploid. Diploid would have to have both. This only has 1, but it has 2 copies of that one chromosome, called a secondary spermatocyte. Now the secondary spermatocyte divides again. So each of these actually, there's a cytoplasmic connection here. Tiny, tiny, tiny one, but still connected by cytoplasm. So now each of these is going to divide again. And we have 4 cells. Still outside the cytoplasm. These are now called spermatids. And finally, we have a completely just N, which is what our sperm is going to have. They still are round cells, and so they are identified as early spermatids, what you see here. Their whole purpose, remember, is to just transport DNA. So they lose a lot of cytoplasm and they grow tails. Okay? So now we see their heads buried in the cell. They look like sperm, but because they're still associated with the Sertoli cell, they're still considered to be spermatid, only now they are late spermatid. Eventually, they become free in the lumen. Once they are free in the lumen and no longer associated with the Sertoli cell, they are identified as a spermatozoa. So structurally, nothing really changed. They're just no longer associated with the Sertoli cell. Yes?
So is each one of those late spermatids coming from the early spermatids?
Yeah,
Like
so
they all have
now there's no more cell division. So an early spermatid will develop into one late spermatid. So here are some images looking at the histology of the testis. So this is low power cross-section of various seminiferous tubules, and all these purple dots that you're seeing are the developing sperm. They look like they're inside the Sertoli cell. Remember, they're separated by the plasma membrane, which is too thin to really resolve. Here would be interstitial cells, or the interstitial cells ablated, all right? This is a little bit better picture because it's in plastic rather than in wax. So let's take a look at what we're looking at. This level of information won't be on Thursday's quiz. We have another quiz next week, which is male reproductive histology. All right, so Thursday's quiz, the 2 questions we have on male will just be the model structures. We'll give you a little bit more time to work on this. So here's our lumen. Here's the connective tissue on the outside of a seminiferous tubule. Here's another cross section. And here are interstitial cells in between. You can see here. So these cells that are right along the edge, down at the bottom, here, 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 those are the spermatogonia, okay, which is plural for spermatogonia. So those are the least differentiated. They're the most like all the rest of your body cells that have a 2N, a diploid chromosome. Then each of these will divide. One will remain behind, and the other will move up towards the lumen. And this is the most distinctive of the cells. See all these little, they're sometimes called bouquet cells, because we can actually see those condensed chromosomes. And these are the primary spermatocytes. Now, do not ever identify a cell in here as a secondary spermatocyte. They are present, but hardly ever captured in histology because they only stay as secondary spermatocytes for a few seconds and they divide right away. So the possibility of capturing them in a histology section is pretty poor. So you'll see them in diagrams as part of the stage when the primary spermatocyte divides. But here, these are early spermatids. So in a light microscope view, you're not actually going to see a secondary spermatocyte because it's just there for a moment and then it divides and forms the early spermatid. Okay? So it does appear, but briefly, yes?
So can we just label the big ones as spermatocyte, or do we have to use, say, a primary?
Primary. Because in essence, they're all spermatocytes, but we just call them spermatids. So I mean, spermatocyte is technically really just a sperm cell. But the primary sperm, so you need to say primary because it's not a secondary. I do want you to recognize it's the first one that appears. So spermatogonium, primary spermatocyte, and then the early spermatid. So you remember from 430 what information you can derive from the fact that the nucleus is small and compact and all heterochromatin? Pardon? That, but more specifically, it's not undergoing protein synthesis. Remember when it's highly coiled, it can't make new proteins because it has to uncoil to form new chromatin. So these were a couple of the cells I used as examples of complete heterochromatin in 430 because they're so condensed. All right. Now they're not really about to divide because early spermatids don't divide again. They're just becoming very condensed so that they can fit all their chromatic material in this tiny little head of the spermatozoa. So that's part of the changes that the cell undergoes. That chromosomal material becomes more and more and more and more compact, and we lose a lot of the cytoplasm except for microtubules that are forming the tail. So notice how these all have their heads angled in this direction? So these are the late spermatids. Okay? So again, spermatogonium divides, one stays behind and forms a primary spermatocyte. That will divide to form the secondary spermatocyte, which we won't see anymore. The secondary spermatocyte divides and forms these early spermatids. So the cells are pretty distinctive. Okay, it's not really difficult to tell the difference between them. And then the early spermatids are really easy to tell from the late spermatids. Here is a nucleus of the sustentacular cell with a nucleolus. All right. Here's another one here. It's hard to tell where the actual lateral membrane is of the cell. You really can't pick that out. Here's another sustentacular nucleus down at the bottom, or two. You may not be able to see those in the back, but if you look at the PowerPoints online, you can see them. Here's part of one. But probably the best ones you can see are this one and this one. Here's another one up here. See how tiny that? So that's the nucleolus, and then this is the nucleus of this whole cell. So this would be a sustentacular cell here. That's the size of the sustentacular Sertoli cell.
What did you say that late spermicides are called once they're in the lumen?
They're called spermatozoa. Okay? Now, another thing that will be on your exam, heads up, as I say this every semester, and so far you guys can be the ones that break the mold. Every semester, at least half the class misses half of this particular question. So, you know ahead of time, there will be a table on the exam in which I ask for the chromosomal pattern of these cells. It's on the board up here, and we will be going over it in detail on Thursday. All right, the identical pattern is seen with egg development.
So do you mean the 2N, 2N,
Yes.
2N, N?
Uh-huh. Okay, so your textbook just says 2N here. And it is 2N, it is a diploid cell, but I add that extra 2 to remind you that everything is actually doubled. Okay? So what would be the chromosomal pattern of an interstitial cell? In reference to N and 2N and so on.
2N?
2N. It's a typical somatic cell. So interstitial cells in Leydig are going to be 2N, just like your muscle cell and epithelial cell lining your mouth. Okay? Just a moment. What would be the chromosomal pattern of the Sertoli cell?
2N.
2N as well. Okay? The only thing that changes are developing gonadal cells. Yes? Question?
Do you want us to write 2N or
For that table, I want you to write 2N or 2 parentheses N or 2 parentheses 2N. And again, on Thursday, we'll go over that. And there's part of your homework also asks you to do that in the next lab report. Okay? All right. So we'll come back and visit this, and we'll do it again with the egg. Because the same types of chromosomal changes occur in the egg. So let's look at the spermatozoa. This cell here is what looked like that early spermatid. So you can see that it does undergo distinctive changes. Because again, remember, its only purpose is to transport this information to the egg. So we'll go over the sex hormone synthesis and all of that on Thursday. I just want to get through some more structures here. So move to page 68 and 70. So we start out with that early spermatid, and then we lose a lot of cytoplasm, and we get this distinctive head, midpiece, and tail. You guys still need pictures of that? If the sperm kept as much cytoplasm as the egg does, you guys would be carrying around testes that look like this, okay? And it's just not needed. It would be difficult to maintain a metabolic activity of a cell that size, and so the egg has all that cytoplasm and the mitochondria and all of that.
Is that what happens with oh shoot, I can't remember now.
There is a
Really?
condition where there's blockage of the lymphatic ducts that leads to elephantiasis.
That's the
Yeah, that's a buildup of lymphatic fluid when the scrotal sacs reach down to the floor if they're sitting on a stool. Yeah, that's a lymphatic fluid blockage. So we develop in the human, the head gets kind of this arrowhead appearance. And within that, we have the nucleus, which has DNA. Remember, it's haploid, so this is going to be half the amount of chromosomal material. Most of the organelles disappear. We no longer need ribosomal ER, all right? The Golgi disappears, except for one of the Golgi cisternae, and that remains kind of like a ski cap, you know, that you would pull down over your head. And it is like a little cap that gets pulled down over the nucleus. So this is one of those things that you would put develops from. This is known as the acrosome or acrosomal sac. And it develops from one of the Golgi cisternae. This contains enzymes that are released within the female reproductive tract and allow the sperm to penetrate through the protein coat that protects the egg. This took a lot of work in developing in vitro fertilization, because just sticking the sperm in a petri dish, the sperm weren't releasing the hydrolytic enzymes, and it took a lot of work, including some scientists over at UC Davis, to develop the proper recipe, if you will, the chemicals and hormones that needed to be present to trigger the release, the membrane fusion and the release of these enzymes. Then we have a mid-piece. And in the mid-piece, we have kind of something like a basal body, the 9 triplets of microtubules. So we have the origins of microtubules. Now, is it microtubules or actin that form motile structures, that form the cilia? Remember, the cilia can beat microtubules, all right? Actin is just a surface area increase, but between two set microtubules are little arms activated by ATP that cause the microtubules to slide back and forth. And so in the respiratory tract, with the cilia can beat and move the mucus up the trachea. That requires energy. That requires ATP. And ATP is produced by mitochondria, all right? So mitochondria are present, circular mitochondria, wrapping around the microtubules. So they have instant access to the ATP. And this powers the single flagellum. So from the mid-piece, we see the presence of the doublets. So these extend down into the flagellum. But in cross-section, remember the 9 doublets? 1, 2, 3, 4, 5, 6, 8. We have this 9 doublet pattern. So this would be a cross section. Through the flagellum. And it doesn't just whip back and forth. Kind of looks like an egg beater. It goes around. So it kind of does this. So it goes in a circle as well as beating and propels the sperm. We don't need any other organelles. All right? So when the sperm fertilizes the egg, this ruptures, releases the enzymes, and the nucleus DNA is propelled into the egg. Now, so this is diagrammed, and we'll go over the fertilization process on page 70. But different sperm from different animals, different species, have different appearing heads. So this kind of arrowhead is the appearance, can I erase this? Is the appearance of the human sperm. It actually comes in sideways. So when it fertilizes the egg, it's here on the side that it ends up binding to the egg. It doesn't, go into it like a missile. So humans are shaped like this arrow. Hamsters, guinea pigs are kind of shaped like a sickle. And pigs are shaped like ping pong rackets, paddles. Humans seems to be more dynamically stable, all right? But they do have these different. So when I was teaching at the med school, my son was 2 or 3, and he was in daycare. I only taught 2 days or 4 afternoons a week, so I had a lot of free time. So I volunteered in one of the sperm labs, one of the guys that worked with in vitro fertilization and figuring out the contents. Our sources for human sperm were mostly medical students, and they would pay them, this was back in the early 90s, mid-90s, $25 an ejaculate. So we were right around the corner from the men's room, and they would have a little urine specimen cup and go there and ejaculate and then bring it to us. Well, I also taught them as their first year medical classes, so I'd be sitting on the bench and they'd walk in with this cup of ejaculate, see me, and turn around and walk right back out again. So I would leave and I'd go into the inner room and they'd come back in, somebody would go get them, and we had a CO2 incubator. We could keep them alive for about 2 weeks and do research with them. The sperm, not the donors. And we would wash them and count the sperm, make sure they didn't have too many extra heads and extra tails and that type of thing. The typical ejaculate is about 2 to 5 mils with about 50 million per mil, so about 250 million total. The volume comes from the prostate gland and seminal vesicles. My favorite donor had about 1 billion because I used the electron micrograph and I did a lot of microscope and I did a lot of washings, and so I needed to see that pellet at the bottom of the centrifuge tube. And so I had tons of sperm. We didn't pay him any extra, but he should have demanded it. One of my colleagues was a graduate student who finished her PhD while I was there, and she worked with pigs and did fluorescent antibodies and light microscope studies with them. And when she graduated, she and the principal investigator tried to get me to take over her work. And I said, I already have my degree. I'm not getting paid. I'm doing this for free. And pigs, unlike cows and sheep, don't respond to electrical ejaculation. They require manual handling. And my friend would often have to go home and take a shower before she came back to the lab. So I was like, I don't think so. Not for free. But it's interesting to see the different shapes. They all end up binding to the egg and fertilizing the egg, but they often have these different overall appearances. So when we come back and talk about fertilization, we'll come back and do the sperm structure. So let's move on. I want to get to some of the other male structures in the next 15 minutes. So here's a view of some of those sperm. We'll talk about the acrosome reaction when we get to fertilization. It was the thought from some people that the entire human being existed in the sperm, just in miniature. And so this was proven one day. You can see the little embryo sitting down here. That's not Photoshop. This happens to be some folding of the sperm membrane. So here it is coming sideways to attach to the egg protein surface. So now let's follow the sperm. So all of this, it doesn't swim out of the testis. It can't swim yet. It can't beat its flagella. It doesn't have the enzymes that are, that's the enzymes, but they're not in the mature form. And so it's actually propelled by the flow of fluid produced by the Sertoli cells. So they secrete a fluid, and that pressure pushes the sperm through the seminiferous tubules and then out the speedy testes and the efferent ductules and into the epididymis. And the epididymis is a single tube. All right, there's only one, but it's highly coiled. So again, when we look at a histology of it, we're going to see multiple cross sections. The cluster up here is known as the head, the body, and then before it straightens out, it's known as the tail. So this is where the sperm resides until ejaculation. Two things happen in the tail, and they both are components of maturation. So in the tail is where the acrosomal enzymes mature. If you were to take sperm from the epididymis and put it in an in vitro dish with an egg and all the proper chemicals and hormones, and if it could swim if the sperm could swim to the egg, it wouldn't be able to penetrate through the protein coat because those enzymes wouldn't be functional. Okay. The other thing that happens is the enzymes that activate the flagella become functional. So now the flagella would beat. So up here, they wouldn't even be able to swim to the egg. All right. And if it got there, it wouldn't be able to penetrate through the protein coat. So those two events, the maturation of the hydrolytic enzymes in the acrosome, and the maturation of the enzymes, also there's a destabilization of the membrane. The maturation of the enzymes that allow the flagella to beat occurs in the tail. Now, a process was developed kind of serendipitously. A researcher wasn't trying to fertilize the egg, but was trying to study the steps of egg activation. Took a very long, you can take glass pipettes and hold them over a Bunsen burner and heat them, and then just stretch them, and the opening of that lumen can become as tiny as a hair follicle, just by stretching that out. And that can be, you can take up a single sperm and push that tiny, tiny lumen of the pipette through the egg membrane and inject the sperm. And the egg will undergo the reactions that it normally does, about 70% of the time. And so this is now developed into a step that can bypass, say, form of in vitro fertilization that can be used when the sperm can't beat. So somebody that has a genetic disorder where they lack the enzymes for the microtubules, or maybe because of genetic problems, they want to specifically choose one sperm to fertilize the egg. Now, when we look at the histology, it looks pretty similar at first, okay? They both have a lot of cross-sections of luminal branches, luminal components. However, if you look closely, all these little dots that are in the testes are there because of the developing spermatocytes, the spermatogonia and the primary spermatocytes and so on. In the epididymis, this is a pseudostratified columnar epithelium. So the nuclei are all going to be at the base. There'll be multiple layers appearing, but they're all at the base. Okay? Now, the edge of the lumen looks irregular because of the tails of the late spermatid. What's creating this irregularity are long microvilli in the pseudostratified columnar epithelium. Remember the pseudostratified columnar and the trachea had cilia? These are very long microvilli. So here's a high-powered view. Notice how there are no nuclei up here? This was the testis. We would have the late spermatid, and the early spermatids, and we have the late spermatids with all their heads embedded in there. These are not tails of late spermatids, but the long microvilli that are clumped together. So when you look at the detail, all right, there is quite a bit of distinction. Also, there's a thicker connective tissue surrounding the vas deferens, the epididymis, and there's no interstitial cells. Okay? So compare this appearance with the testis. Even at a low power view, you can see how it's completely filled with the nuclei. But again, I'll give you a couple of lab days to work on this with the microscope. We won't have histology on Thursday's quiz. All right, then we move into the vas deferens. This is just a view of sperm aspiration from the epididymis. All right, where there's a higher concentration, it only moves into the vas deferens during emission. So prior to that, they can go into the epididymis and take sperm. This is done sometimes with individuals, men who are paralyzed, all right, and can't have an ejaculation or emission, so they can go in here directly and remove the sperm. So we've done the testis, epididymis, and now we move into the vas deferens or the ductus deferens. We're going to do that and the seminal vesicle, and then we'll stop. We'll do the urethra on Thursday. Vas deferens is also a single structure. So I have this image in your lecture notes. It's not highly coiled. You're not going to see a whole bunch of multiple circles. They're smooth muscle now. All right. This is pseudostratified. The only other thing you might confuse it with would be the urethra, which is also a single structure and has smooth muscle around. The urethra is the tube that carries urine and or sperm from the bladder to the external opening of the penis. Anybody remember from histology last semester what the epithelium lining the urinary tract is?
Simple spins.
Not simple.
Cuboid.
It was the one that changes?
Stratified?
No. Sometimes it's multiple layers
Cuboidal?
that look like tiers. The top layer can either be cuboidal or a flat cuboidal, depending on whether it's stretched or not.
Stratified cuboidal?
Transitional, remember the term? It transitions. So it can go from 15 layers to 3 layers. And it can go from simple cuboidal to flat, not quite squamous, but almost. Some of the cells can have multiple nuclei. It's the one that was looked, you could confuse it with stratified squamous. Stratified squamous is the horizontal layers, and transitional is vertical layers. The big round biscuit-shaped cells on the surface if it's relaxed. So, pseudostratified columnar is pretty distinctive from transitional. So if you look, can see the actual epithelial tissue, that's going to be the distinction between those two. Otherwise, you're not going to confuse them. For birth control, all right, vasectomy, the vas deferens is the tube that is cut in the male and blocked or tied off. It's not done in the pelvis like the oviduct is in the female, but it's done through the scrotal wall. Okay. After we had the twins, we only planned on having 2 kids, so after the twins were born and we had 3, we were done with our family. And I kept asking my husband to have a vasectomy, and he kept putting it off. So I was on birth control pills, I had the IUD, and I was like, okay honey, this is enough of this. I said, he doesn't like needles. He tends to faint when his blood gets drawn. I said, if you just don't want a vasectomy for the principle of it, I'll back off. But because you're afraid of the needles, I'm going to keep bugging you about this. Because he had promised me that he and his best friend were going to go and get a vasectomy together and sit there and hold hands and have a bag of ice over their scrotum and so on. And it wasn't happening. The twins were probably about 7 or 8 years old, and he came home with a form for me to sign. He had gone to his doctor and attended this group class where they were all told about how the vasectomy was performed and the pros and the cons. And I had to sign it before the surgeon would perform the vasectomy. I said, you're kidding. Our daughter, when she's a teenager, can go and get an abortion without me even knowing about it. But for you to get a vasectomy, I have to sign that you've told me you're going to do a vasectomy. It just seemed a weird balance of things. So there are a couple of different ways. One is actually making an incision. And the other, which was developed in Japan, involves poking a hole, kind of tearing the tissue. It's supposed to be less painful and heal faster, but it doesn't sound. So it'd be quicker to just cut it. But they palpate for the spermatic cord. Obviously, you don't want to cut the artery or the nerve, because then there go the sperm. They inject a local anesthetic and find the vas deferens, bring it out, cut off a segment. And now they can do a couple of different things. All right? They want to remove enough so that when the elastic seed kind of pulls back in, it's not going to reattach, because that can happen. And there were quite a lot of divorces where women got pregnant and their husbands accused them of sleeping around. This is before you could do a paternity DNA test. And they swore they didn't, and the guy's going, well, I had it cut, so it's not me, when it really was. So they will bend the edges over, clamp them, suture them. They can put a piece of plastic cylinder in there to block it. That makes it easier to reattach them, but if you even say, can I get it reattached in 10 years, they probably won't do the surgery, because it's supposed to be assumed to be permanent, not, well, I don't want to get, I'm going to do this for about 5 years, 10 years, and then I'm going to divorce, or I'm going to, what happens? I'm single now, I don't want to worry about birth control, but I might get married, and then I want kids. So don't go into that kind of thing. It's supposed to be considered to be permanent. This is the no scalpel method, where they kind of pinch it through with a caliper, and then they just kind of tear it through. And to me, that doesn't sound like it would make it heal any faster, but apparently it does. Okay, you just kind of pull it through. Make sure it doesn't have a pulse,
This is traumatizing.
so it's not an artery. And then cut it, walk it, whatever, and let it pull itself back in again. These guys, including my husband, are still making sperm. All right, they're still producing testosterone. The testosterone is an endocrine chemical, so it's still getting into the blood and going to the rest of the body. They still have an ejaculate, all right? Because most of the volume is coming from these glands. So they still have the ejaculate, it just doesn't have the sperm in it. So after a vasectomy, you're not supposed to have intercourse without protection for about 6 weeks, and then have the ejaculate observed in the microscope to make sure it doesn't have sperm in it before you actually resume intercourse without protection. So what happens to the sperm?
Does
They
that
die.
no, it goes back up.
They die in the epididymis and get reabsorbed and broken down and taken back into the body tissue.
Wasn't there an argument about that causing prostate and testicular cancer?
That would be from breakdown products and testosterone. Testosterone can increase the size and activity of the cells, but they don't go back into, they're broken down within the epididymis into proteins and amino acids and so on, and are taken back up. Now that can happen sometimes anyway, and the guys can produce antibodies. Sometimes it's rare because these are their own proteins. It's more common for the woman to produce antibodies. Okay? So that blockage in surgery typically occurs right here along the back side there. If the surgery isn't present. Anyway, so the guy shouldn't feel any difference in ejaculation or in sex drive, because that's not interfered with. Having a woman's oviduct tied off just prevents the egg is still going to be released. The sperm is going to get to that parental blockage and not be able to get through unless it's pretty wily. So this passes through the inguinal canal along with the nerves and blood vessels, and then we see it laterally along the bladder. And again, this is emission, where contraction of smooth muscle of the vas deferens moves it through. So now we're looking at the posterior view of the bladder. Here's the ureter coming down from the kidney. The vas deferens is medial to that. It dilates to form a structure known as the ampulla. And then we pick up the first of primarily 3 sets of glands. These are known as the seminal vesicles. Okay, we'll pay attention to their histology on Thursday. But they produce a fluid that is rich in the fuel source for the sperm. So, citric acid. Remember, well, you haven't had microbiology maybe, or we haven't done it, but glucose is the most common source of carbohydrate. And citric acid is one of the steps after glucose is broken down into 2 carbon groups. And so citric acid and fructose are in high levels in seminal vesicle fluid, along with chemicals that protect the sperm. So antibodies, the more antibodies are actually present in the prostate gland. There's a clotting agent identical to what we have in blood. So you may or may not have noticed, but after sperm is ejaculated, in the first couple of minutes it thickens and becomes more jelly-like, and then it becomes watery. Okay? When this occurs in the reproductive tract, that thickening or gelling is due to the fibrinogen. The resistance to the sperm swimming gets them oriented all with the heads pointing in the same direction, like a sea of minnows, group of minnows. And then the fibrinolysin breaks down the fibrinogen and they're free to swim. And most of them head up the reproductive tract instead of swimming away in the opposite direction. So that's due to the fibrinogen that we see. And some bicarbonate ions that neutralize the acidity of the urethra. Okay, and the vagina as well. The sperm and the seminal vesicles join together to form a common tube that passes through the prostate gland. This is identified as the ejaculatory duct. So the ejaculatory duct is formed by the union of the prostate gland and the seminal vesicle. And you can see one of them right here. Okay, so that's the ejaculatory duct. And it opens into this segment of the urethra that's surrounded by the prostate gland. The prostate gland is kind of like a donut. Prostate gland has multiple openings. It has more of a milky fluid. And we'll come look at this histology on Thursday. So here's the prostate gland, 2 openings for the ejaculatory duct, but multiple openings for the prostate. This is where prostaglandins were originally thought to only come from. And they come from the seminal vesicles as well. This milky fluid also adjusts the pH, has antibiotics, and more of those clotting agents. So primarily the volume of semen is due to the seminal vesicle and prostate gland. The sperm itself contributes probably about half a mil or so of the 2 to 5 mils of volume. All right, so let's stop there. We don't have enough models for every single person to have their own models, so the models are in halves. They can separate, so share them among the tables. If you want to get started on the histology, you may, or if you want to go back and work on the histology for the endocrine system. So your quiz on Thursday will be 4 questions from endocrine models and histology, 2 questions from male reproduction, so identifying the parts of the inguinal canal, identifying the parts of the testis, so the seminiferous tubules, straight tubules, and so on, identifying seminal vesicle, ejaculatory duct, prostate gland, vas deferens, epididymis. I won't have you identify the histology of any of those things. I won't ask you to identify primary spermatocytes or early spermatids yet, because that's all in slides. You're certainly welcome to work on them, but that will be in our second quiz. Okay? So you can take a break and then get started on lab. I'll pull out the models. Called the bulbourethral or Cowper's glands. That secretes a fluid. That secretes a fluid that helps to flush out any residual urine and lubricate the glans penis, the outside of the penis. So that's this small one right here. Okay. It can be called Cowper's glands. A similar gland in the female shares the name bulbourethral. So those are interchangeable. And And now I'll just leave us with the penis structures to cover on Thursday.